Now that we've had the chance to talk about different types of tissues, I'd like to talk a little bit about glands. A gland can be a cell, a group of cells, or a whole organ, but its defining feature is that a gland releases a secretion. Glands derive from epithelial tissue. It's epithelial cells that are actually releasing the secretion, whether it's a single cell like a goblet cell, which releases mucus, or whether it's a whole organ. When we are talking about a whole organ, this organ is formed by an invagination or folding in of epithelial tissue that creates a pocket of epithelial tissue beneath the surface of an organ or an opening. This epithelial tissue is referred to as the parenchyma of a gland. Around the parenchyma we have what's called the stroma, which is connective tissue that helps to support the epithelial tissue of the gland. There are two main types of glands that differ based on how they release their secretion. Endocrine glands are pockets of epithelial cells that release their secretion into the bloodstream. There's no duct or tunnel that allows it to go out onto a surface, so instead the secretion has to be released where it gets picked up by capillaries into the bloodstream to be carried to different parts of the body. Some examples of endocrine glands would be the pancreas, which releases insulin and glucagon, or the thyroid gland releasing thyroid hormone, or the pituitary gland releasing all sorts of different hormones into the blood. An exocrine gland maintains a tunnel to the surface of the epithelium, and this tunnel is the duct through which the secretion can flow onto the surface or into the cavity. A couple common examples of exocrine glands include sweat glands, which release sweat, or mammary glands, which release milk, or salivary glands that release the saliva into the mouth. We'll talk more about the endocrine glands when we talk about the endocrine system, so I want to focus a little bit on the exocrine glands. Again, these are glands that have a duct so that they release their secretion onto a surface or into a cavity. Exocrine glands come in many different shapes, but we don't need to worry about the details of naming the different shapes of exocrine glands. I want to focus instead on the type of secretion that's released and on the method of secretion. There are three main types of secretions released by exocrine glands. The first is serous. A serous secretion is a watery sort of secretion. This watery secretion can contain all sorts of other substances like proteins, fats, salts, or other molecules, but it's a watery sort of secretion. Serous secretions include sweat, milk, tears, saliva, and other digestive juices, and the fluids that help organs move smoothly against each other, either in the pericardial cavity of the heart, the pleural cavity of the lungs, or the abdominal and pelvic cavities. The second type of exocrine gland secretion is a mucus secretion. A mucus secretion is a thick secretion that contains the protein mucin. Goblet cells are an example of cells that can release a mucus secretion. We also have cells that can release a mucus secretion in the salivary glands. The last type of secretion is cytogenic. This one is interesting because it's the secretion of a whole cell. There are only two glands that release cytogenic secretion. Those are the testes that release sperm and the ovaries that release the ova or eggs. There are different methods of secretion or different ways that these epithelial cells are able to release the secretions that are going to travel down the duct into the opening or the cavity or onto the surface. The most common method of secretion is merocrine secretion. In merocrine secretion, the components of the secretion are packaged into vesicles and then released by exocytosis. This is how the pancreas releases insulin and glucagon, this is how tears are produced, and this is how the gastric glands in the stomach release stomach secretions. Another type of secretion is holocrine secretion. In holocrine secretion, the whole cell is broken down and released as part of the secretion. This requires continual replacement of the cells because instead of just releasing some secretion from a cell, which can then make more secretion, the entire cell disintegrates and becomes the secretion. So we have to have a constant supply of new cells to replace those that are disintegrating. This adds a lot of additional components to the secretion, including a lot of lipids, because all of the lipids that were part of the membranes are now being released as part of the secretion. The sebaceous glands, the oil glands of the skin, release their secretion through the holocrine method. The last secretion method is called apocrine secretion. In apocrine secretion, the components to be secreted go to the apical, or the top surface of the cell, and then the top surface pinches off, and that's what becomes the secretion. Again, because this includes membrane components in the secretion, this secretion has more lipids or more fat in it. The cells of the mammary glands produce milk through the process of apocrine secretion.